Okay, so welcome back to this next video in which we are discussing the heterotrimeric G proteins. Okay, so now what we want to discuss is what's known as the G protein cycle. So this is how G proteins are activated, how they interact with targets, and then how they deactivate. Okay, so the G protein cycle then. Right, so we're going to start off with our off heterotrimeric G protein, which is the G protein where the alpha subunit has guanosine diphosphate bound to it and is therefore in the off state. And in this off state, it will associate with the beta gamma complex here. Okay, now we've got our G protein coupled receptor. So if I just extend this membrane a little bit, we have our G protein coupled receptor, which has these 7 membrane spanning alpha helices here. And this currently has no ligand bound to it, so this is a GPCR, a G protein coupled receptor. Right, okay, and we'll keep this completely general. We won't say which family it's in, we won't say specifically which one it is. Okay, so basically, when the ligand binds to the G protein coupled receptor, that will trigger a conformational change that allows the G protein coupled receptor to bind to the heterotrimeric G protein. Okay, so let's draw this as well we can. Okay, so here is our heterotrimeric G protein. Okay, 1, 2, 3, 4, 5, 6, 7 there, and now some ligand has come along and bound to this G protein coupled receptor. And what it does is it allows a change in these intracellular loops here. Okay, so the intracellular loops are named intracellular loop 1, which is this first one between the first membrane spanning alpha helix and the second, intracellular loop 2, which is this intracellular loop between the third and fourth membrane spanning alpha helix, and intracellular loop 3, which is between the fifth and sixth membrane spanning alpha helix. Okay, and when the ligand binds, these will undergo a conformational change, and now what will be able to happen is that the intracellular loops will have a binding domain for the heterotrimeric G protein. So now I'll show the heterotrimeric G protein binding with the G protein coupled receptor, and it's going to have to get a little bit more squashed in, basically, to fit in. Okay, so here are our 2 main portions, the beta gamma complex and the alpha subunit. And I'll try to put the lipid molecules still sort of getting into the membrane. Okay, so here is our palmitoyl or myristoyl group. Okay, and here is our prenyl group coming off the gamma subunit. Okay, here is the gamma subunit in blue here. Here is our beta subunit in turquoise here. Here in red is our alpha subunit, and of course it still has guanosine diphosphate bound to it at this stage. Okay, so here, within here is GDP, and I'm not going to be able to actually fit the GDP in there, but in there should still be the guanosine diphosphate. Now, the guanosine diphosphate isn't actually going to stay in there for very long. Basically, once the heterotrimeric G protein binds to the intracellular aspect of this G protein coupled receptor, and as I say, it's these 3 intracellular loops that make the binding domain for the heterotrimeric G protein. What happens is that the guanosine diphosphate is induced to leave, okay? So the alpha subunit loses affinity for binding to guanosine diphosphate, and another molecule in the cytoplasm of GTP, this time guanosine triphosphate, will come in and it will now bind to the alpha subunit. So once the heterotrimeric G protein has bound to the G protein coupled receptor, guanosine diphosphate goes off and guanosine triphosphate comes in. Okay, now when the alpha subunit has GTP bound to it, it goes into the on state. Okay, so we are now in the on state. Okay, so you've basically switched the G protein on. Okay, and when the alpha subunit has GTP bound to it and is in the on state, it no longer wants to associate with our beta gamma complex over here, so the 2 will separate apart. Okay, so let's now draw this here. Okay, so we have our G protein coupled receptor now still in the middle. It's done its job now, so we're not really interested in that anymore. And the beta gamma subunit goes off over here. Okay, so here it is. Like so, the gamma subunit still with its prenyl group, which is attaching the whole complex to the inner leaflet of the phospholipid bilayer. Okay, so here's the gamma subunit in blue, the beta subunit in turquoise, and the prenyl group coming off the gamma subunit that anchors the whole complex into the inner leaflet of the lipid bilayer. And then, of course, the alpha subunit also goes off here, so I'll draw it a little bit bigger so that I can actually fit the GTP molecule in the middle. Okay, here is the GTP in the middle, and then I'll colour in the alpha subunit that is now in the on state in red still. Okay, and of course it still has its acyl group, whether that's a myristoyl group or a palmitoyl group, or potentially you have two, and that is anchoring it in the inner leaflet of the lipid bilayer. Now what will happen is these two subunits will go off and interact with targets. Okay, so let's say the beta gamma subunit has some target over here, and this is an important concept. For a very, very long time, people thought that it was just the alpha subunit that actually had important signaling roles, but actually the beta gamma subunit also has important signaling roles. Okay, so here's a target for the alpha subunit over here, target 1, but there is also a target over here for the beta gamma subunit. And what will happen is the two subunits will go and bind to their targets, so the alpha subunit will go and bind to its target, and the beta gamma complex will go and bind to its target. And of course they can have multiple targets which they can all be binding to. Now basically, when the alpha subunit binds to its target, what happens is the target activates the alpha subunit, and the alpha subunit then is going to chop up the GTP, and it's going to break it down into GDP and inorganic phosphate. So basically, alpha subunits of heterotrimeric G proteins are GTPases. They are enzymes. They have within them the enzyme that is able to split GTP molecules back into GDP molecules and inorganic phosphate. Okay, so alpha subunits can take GTP molecules and break them down into GDP molecules and inorganic phosphate molecules. Now, if you just leave an alpha subunit with GTP, it will bound to it. It will eventually break the GTP down into GDP and inorganic phosphate, and it will eventually hence turn itself off. However, when it interacts with its target, obviously it will activate the target potentially. But also, what the target will do is it will activate the alpha subunit as well, and it will speed up the rate at which the alpha subunit actually breaks the GTP down into GDP and inorganic phosphate. And different targets will have differing abilities to activate the alpha subunit and get it to turn itself off, basically. So some targets will make the alpha subunit turn itself off extremely rapidly, and other targets will take much longer to force the alpha subunit to turn itself off. Okay, so what happens when the alpha subunit turns its GTP to GDP and inorganic phosphate is that the inorganic phosphate will be released out into the cytoplasm, and the GDP will remain bound to the alpha subunit. Now, once the alpha subunit has GDP bound again, it will cleave away from its target, because only the alpha subunit with GTP bound can interact with its target. So it will cleave away from its target. So let's draw this. Okay, so here is target 1, the target for the alpha subunit. And now as soon as the alpha subunit has cleaved that GTP to GDP, then basically it will dissociate away from its target, basically. It can't bind to it anymore. As soon as the GTP has gone and it's got GTP instead, it can't bind anymore. Okay, so the inorganic phosphate then goes off into the cytoplasm. Like so. Okay, so let's just colour in the little essential bits here. Okay, so here is the acyl group, and here is the alpha subunit again in red. And then what will happen is the alpha subunit with GDP bound to it will then have affinity for the beta and the gamma subunit, and it will basically go and nick the beta gamma subunit off the target for the beta gamma subunit. So basically it will take the beta gamma subunit back now, so it's quite a horrible molecule really. It rejects the beta gamma complex, and then later, once the beta gamma complex has found someone new, it comes and steals the beta gamma complex back again. Okay, so here is the beta gamma complex with the prenyl group attached to the gamma subunit, and basically you've now got the off G protein again, and hence we've gone round in a cycle. We're back to where we started, and this basically is the G protein cycle, because now this is the heterotrimeric G protein back again, which can interact with the G protein coupled receptor again if the ligand comes along again. Okay, so that concludes our discussion of the heterotrimeric G proteins.